So now we are starting on Unit 3. In Unit 3, we're going to be talking about a couple different types of cultures from different specimens. We're going to be talking about CSF, urine, and GI or stool cultures, and the pathogens associated with those types of cultures. And then we're also going to talk about the group or family of organisms known as the Enterobacteriaceae. So this first lecture from Unit 3 is going to cover just CSF and urine cultures. Okay, so CSF, which is also called cerebrospinal fluid, just some general information. So this is the fluid that covers the brain and spinal cord. It cushions the brain and transports needed metabolites and waste products. So the idea is that only certain things are supposed to cross the blood-brain barrier. So when things that get in there that aren't supposed to be there, this is when, infections occur, which can be fatal. Cerebrospinal fluid is normally clear, colorless, and sterile. Other colors or turbidity may be an indication of infection. And then some host risk factors. So this includes extremes of age, those infants or very young or very old folks whose immune systems are not as robust, nutritional and immunologic status, comorbidities such as alcoholism, diabetes mellitus, some type of malignancy, renal failure, head trauma. So any of those other conditions that could be going on at the same time, again, and this just kind of contributes to the overall host immune status. And then if the patient has undergone any recent neurological procedures. Okay, CSF testing and culture. So the CSF, the actual spinal fluid, is collected by aseptically inserting a needle into the subarachnoid space in the spine and then withdrawing the fluid. That procedure is referred to as a lumbar puncture, or you probably have heard it referred to as a spinal tap. Usually the total collection volume is going to be around 10 mLs, and that's collected in either 3 or 4 tubes, and that's kind of decided on by the facility whether they use a 3 or 4 tube collection system. And depending on whether it was a 3 or 4 tube collection, you could use either the second or the third tube for your microbiology testing. And you want to centrifuge that sample and use the button for your testing. Never refrigerate the specimen, and that's just because that some of the organisms commonly recovered from CSF are extremely sensitive to temperature. And then the media that you would set that culture up on would be your blood agar plate, chocolate, and then your enriched broth or your thio. And the direct smear or the gram stain, you have to do this on a cytocentrifuged sample. And, as I mentioned this previously, I think, CSF, it should not contain anything. And even a lot of times when it does contain something, it's the quantity is so small, so we have to cytocentrifuge that specimen, which just gives us a very concentrated specimen to view and to work with. It just increases the likelihood that we're going to recover or detect any organisms that might be present. If we do either recover an organism through molecular testing, or we have a positive gram stain, and I don't mean gram positive, I mean we detect an organism on the gram stain, that's considered an alert or a critical value because CSF is a sterile fluid. There should not be any bacteria in a CSF, so we would have to call that result to the physician or to the nurse. And here's the collection tubes that are commonly used for CSF specimens, and you can't really see the numbers on the tubes themselves, but the tubes are labeled, and the spinal fluid should be collected, distributed in those tubes in that order, and then delivered to us. And we take, you can kind of see the gradations on the side of the tubes there. So we determine the total volume that was collected, and we have to notate that on the report. Okay, this is a cytocentrifuge. So it's just a centrifuge method that, you've got this special type of centrifuge here, but then you've got this special like funnel and filter paper type contraption here that you end up putting a microscopic slide behind here, and what's not in the picture is kind of like the holder that keeps all of this stuff together. And then you would drop your specimen, a few drops of your specimen into that funnel, and then as it spins, you can kind of see the little tube there at the bottom. All of the sample goes through that tube and concentrates into that, you see the circle there, it goes through that hole and concentrates in that one little circular portion on the slide. Okay, some different types of routes of infection. So this can occur in the bloodstream via the blood vessels of the brain. So this is the most common way that this happens. It can occur through direct spread from an infected site close to the central nervous system. So that could be like upper respiratory tract infections like otitis media, sinusitis, or mastoiditis. So there's another infection in the general area that allows that infection to make its way to the central nervous system. Some type of anatomic defect in the CNS structures, and that could be due to trauma, some type of recent surgery, a congenital abnormality. And then infection can travel along the nerves leading to the brain. So this is the least common route of exposure. It occurs with organisms such as the rabies virus that travel along the peripheral sensory nerves. So just a few different ways that CNS infections can occur. This can also, infections can be caused by bacteria, virus, fungi, spirochetes, or parasites. Any of those can be life-threatening. So meningitis is the more common type of CNS infection. So it involves the meninges, is more commonly of bacterial origin and usually has an acute onset. So some of the symptoms associated with meningitis include headaches, stiff neck, vomiting, photophobia, which once again means sensitivity to light, and altered mental status. And then encephalitis is when it's still an infection of the CNS, but there's involvement with the cerebral cortex. And the more common cause of encephalitis is viral origin. So symptoms with encephalitis could include seizures and rashes that are secondary to the viral origin. So we're going to focus more on meningitis since they're more commonly of bacterial origin. And here's just kind of a visual representation of the difference between meningitis and encephalitis. Okay, bacterial meningitis. And there should be a list, we're going to go over some of them, but there should also be a list of etiologic agents for bacterial meningitis in the book, and they relate it to age. 95% of all meningitis occurs in children under the age of 5. CSF associated with bacterial meningitis is going to have increased neutrophils, so these are a type of white blood cell. And if that white cell count is high enough, then the sample would be turbid or cloudy. You're going to see decreased glucose and increased protein. So we perform testing on spinal fluids. If we get a spinal fluid sample into the lab, we're going to do microbiology testing, chemistry testing, and hematology testing. So hematology is associated with that white cell and red cell count and the different types of white cells that are present in the sample. Chemistry is going to be your glucose and your protein results, and then the culture and gram stain that we would do in the microbiology department. So bacterial pathogens associated with neonates. So they define that as less than 1 month old, where the patient's going to acquire the organism during passage through the vagina, and therefore the organisms associated with this type of meningitis are different than in other age groups. Meningitis in neonates has a mortality rate of 20%. So group B strep gram-negative rods such as E. coli, Klebsiella, and Enterobacter, and then Listeria monocytogenes. So those are the ones pathogens mainly associated with meningitis in neonates. In infants, which they define as between 1 and 23 months, common pathogens include E. coli, Group B strep, Haemophilus influenza, Streptococcus pneumoniae, and Neisseria meningitidis. In children over 2 years of age and adults, Streptococcus pneumoniae and Neisseria meningitidis are the most commonly recovered etiologic agents. And then older adults, which they define as greater than 65 years, Strep pneumoniae, Neisseria meningitidis, Listeria monocytogenes, and then aerobic gram-negative rods. Back up. Okay, and here is a gram stain that was done from a CSF sample. So you can see this is a white cell. This is a white cell. All of these little small things in the background here are bacteria. They're gram-negative rods. So that's just an example of what a gram stain from a spinal fluid might look like. Okay, viral encephalitis, which is sometimes referred to as aseptic meningitis. Your lab chemistries, so viruses are going to be the most frequent cause of aseptic meningitis, and this is just a milder form of meningitis. Your lab chemistries would show you're still going to have an increased white cell count, but as with bacterial meningitis, you're going to have a predominance of neutrophils. With viral, you might have a predominance of neutrophils early in the early stages of the disease, and lymphocytes, which are a different kind of white blood cell, later on. You could have normal to increased protein and normal glucose results, and this type of infection usually is going to resolve on its own. Patient might have flu-like symptoms, but it'll just kind of resolve on its own, especially in healthy individuals. Okay, pathogens associated with viral meningitis. Your primary agents are the enteroviruses. So this is a group of viruses that includes Coxsackie virus, A and B, polioviruses, arboviruses, Eastern and Western equine encephalitis, and others. It also includes the herpes viruses, which that group includes CMV, Epstein-Barr, varicella zoster, which is chickenpox, the virus that causes chickenpox, and herpes simplex 1 and 2. Less common causes of viral meningitis are the mumps virus, lymphocytic choriomeningitis virus, abbreviated as LCMV, and HIV. Okay, you can also have parasitic forms of meningitis, although this is uncommon. Some of those pathogens that are known to cause, parasitic pathogens known to cause meningitis. So I've divided it up here between the free-living amoeba forms and the parasitic worms. So free-living amoeba, you've got Naegleria fowleri. This is a very rapidly progressing and almost always fatal infection. So this organism can be found in warm, fresh water, moist soil. Most cases have been associated with swimming in warm natural bodies of stagnant freshwater ponds and lakes. And I remember it's been a little while, but maybe within the last decade there was a case in, I think it was in Ohio. A patient had gone swimming in just a natural body of water and ended up with an Naegleria infection. Acanthamoeba species, these organisms are found in soil, fresh and brackish water, and sewage. And then Balamuthia mandrillaris, no environmental sources identified, so I'm not sure how sure the patient would come in contact with those. And then parasitic worm forms that can cause meningitis, Angiostrongylus cantonensis. This is common in some parts of the world like Thailand, Malaysia, Vietnam. Infection occurs by ingestion of green leafy vegetables that have been contaminated with snails or slugs that contain the larva of the parasite. Plasmodium falciparum. When it's due to this organism, it's referred to as cerebral malaria, and it happens through the bite of an infected mosquito. So Plasmodium falciparum is the organism that causes malaria. characterized by changes in mental status. Patient may have seizures, motor deficits. It could lead to coma. And then Toxoplasma gondii. This one is acquired through eating raw, undercooked meat or contact with cats, and organ transplants. And just FYI, this organism is the reason why they say pregnant women should not change the litter box. Okay, there's what Naegleria fowleri, the organism looks like, parasitic organism. So these are white cells, these are neutrophils, these are lymphocytes, but then, compared to the size of the white cells, you can see the size of the amoeba there. Okay, fungal meningitis. This one also is uncommon, seen in immunocompromised patients, transplant patients, diabetics. So some of the pathogens that can cause fungal meningitis, Cryptococcus neoformans, which is a type of encapsulated yeast. This is more common in AIDS patients, and we use a stain called an India ink preparation, so it's kind of similar to the Gram stain, but we can use India ink to identify Cryptococcus. Coccidioides immitis. Infections are limited to endemic regions, so those are mainly in the southwest United States, Mexico, Central and South America, and that organism is associated with chronic meningitis. Histoplasma capsulatum, mainly seen in patients with invasive or disseminated candidiasis. So disseminated just means that it's spread throughout the body. So they've had, and remember, candidiasis is a type of yeast. So they've had a yeast infection that has become systemic. And this one also is endemic to the Mississippi and Ohio River basins. Blastomyces dermatitis, acquired through inhalation of Canidia, which is just kind of one of the fungal structures of the organism. It results in a pulmonary infection, which means lung involvement, spreads to the central nervous system. This one is endemic to the Mississippi and Ohio River basins. And then Candida species, which includes Candida albicans, Candida tropicalis, and parapsilosis, causes meningitis and cerebral abscesses. This one can be acquired as a nosocomial infection in patients with indwelling catheters or those on antimicrobial therapy. Okay, here's your picture of Cryptococcus neoformans with an India ink stain. Okay, mycobacterial encephalitis. So mycobacteria enter the body through respiratory droplets. They multiply and spread via the blood. Mycobacteria are acid-fast bacilli. They have a very thick cell wall containing waxes and phospholipids. Acid-fast bacteria retain dye when they're treated with mineral acid or acid alcohol solution. And I don't know if you remember from one of the previous lessons, we talked about the acid-fast stain. Mycobacteria, the organism, they're very, very slow growers. So where the culture can take up to 6 weeks, we can get that acid-fast stain out fairly quickly. Primary pathogens would be Mycobacterium tuberculosis, where HIV is a risk factor. And then a couple other species, Mycobacterium avium and bovis. And then spirochetal meningitis. Spirochetes, if you remember from the very first lesson, they're slender, flexible, spiral-shaped bacteria. Pathogens with this one, Treponema pallidum, and if you remember from the last unit, that's the causative agent of syphilis. Many cases of neurosyphilis are seen in patients with HIV. And then Borrelia burgdorferi is the causative agent of Lyme's disease. CNS involvement is usually seen in patients with early disseminated disease, less likely during late disease. Okay, and here's a picture of an acid-fast stain, Mycobacterium tuberculosis, on an AFB stain. So this is what those acid-fast organisms look like. And then you can see some organisms here that are non-acid fast. Here's a picture. Here's a picture of Treponema pallidum. So those little squiggly organisms. Okay, and we'll go through a quick review here. Normal spinal fluid should be clear and colorless. Procedure for collecting a spinal fluid. It's called a lumbar puncture. Most common causative agent of meningitis in neonates. Group B strep, which is why we screen pregnant ladies during pregnancy for Group B infections. And then what results would you expect to see in a patient with bacterial meningitis? So bacterial, and again, the combination of all of these results together is how we're going to differentiate between bacterial, viral, tubercular, etc. So with bacterial, you're going to see increased protein, decreased glucose, a predominance of neutrophils on the differential, and then all cases would have an increased white cell count. Okay, this next section of the Unit 3 lecture is going to be on urine cultures and UTIs. So urinary tract infections, the pathogens that can cause them, and then how we would process these specimens. So general, well, and here's just an informative slide, an image that shows you kind of the anatomy involved in the urinary tract. So upper, it's divided into upper and lower, kind of what we refer to the respiratory tract as, and then the diseases associated with upper and lower urinary tract. Okay, some general information. UTIs are one of the most common bacterial infections in the United States, and it's the most common nosocomial infection, and this is mainly due to catheterization. UTI is the most common source of bacteremia, so bacteremia is usually secondary, and remember, bacteremia just refers to bacteria in the blood. It's usually secondary to some other type of infection. So UTI is the most common source, primary infection associated with bacteremia. UTIs are more common in women than in men, and this is because women have a shorter urethra and prostate fluid has bactericidal properties. However, as men age and the prostate enlarges and interferes with emptying the bladder, so this is usually around 60 years old, men will start having more UTIs. A UTI is diagnosed based on a combination of symptoms, the urinalysis results, and the urine culture. And the major organs involved are the kidneys, bladder, ureters, urethra, and the prostate. So patient populations that would be at risk for UTI include elderly and children. So again, they just have inherently immunocompromised systems. They either, the children don't have fully developed immune systems yet, and then as we age, our immune system starts to deteriorate to a degree. And then also, a lot of times these patient populations are unable to care for themselves. Pregnant women also who are going through hormonal changes, you've got an enlarged uterus that puts pressure on the bladder and can impair urinary flow. Catheterization, so it depends on the duration of the catheterization. Has catheter care been appropriate? And then also just host susceptibility. Kidney transplant recipients. So again, this patient population is going to be immunosuppressed. They've got a foreign body that has been implanted or transplanted to their body. So their bodies are going to see that as foreign and are going to try to fight it off. And there's also scar tissue involved with transplant. Patients with spinal cord injuries, and then patients with some type of genitourinary tract abnormality. Okay, the diseases, so it's determined by the anatomy of the organs involved. So again, this includes the urethra, bladder, ureters, prostate, and kidneys. So it can be divided into upper and lower UTI. And you can look back at that diagram also. So pyelonephritis is an infection of the kidney. So this is upper. Cystitis, inflammation of the bladder, that's considered lower. Urethritis is an inflammation of the urethra, that is lower. Prostatitis, infection in males involving the prostate, that would be considered lower. Cervicitis, inflammation of the cervix, that one is lower also. And then bacteriuria is just a term that refers to the presence of detectable bacteria in the urine. Okay, a few different specimen types that you may receive in the lab. And just a note, preventing contamination during the sample collection is crucial in determining the presence and proper treatment of a UTI. But then again, you would kind of incorporate a few different results. So your urinalysis results in conjunction with patient symptoms and culture results. So a voided midstream clean catch, this is the most common collection type. It's considered a non-sterile specimen because it can be contaminated with bacteria from the urethra unless the first portion of the voided specimen is discarded. But this is a non-invasive method of collection, so usually this is what we're going to ask the patients to collect for us. A catheterized specimen is considered invasive, but it reduces the risk of contamination. Contamination is still possible, though, since the catheter passes through the urethra. And then suprapubic aspirate. This one is sterile, where you've got a needle that goes through the belly, into the abdominal cavity, and into the bladder. So this is done when there's some sort of obstruction, maybe in pediatric patients. So this is the only urine specimen type that would be appropriate for anaerobic culture. Okay, a little information about specimen handling. So the specimen should be refrigerated immediately or delivered to the lab within 30 minutes. Ideally, we want the specimen processed within 2 hours after collection. Bacterial counts in refrigerated specimens remain constant for up to 24 hours. And there are also preservatives that can be used to keep the bacteria viable without causing them to replicate for as long as 72 hours. But, again, best practice is just to get the sample and plate it to the media as soon as possible. So culture setup and testing. There's a rapid screen available called a nitrate reductase test or a grease test that tests for bacteria that would be capable of reducing nitrate to nitrite. And there's also one called leukocyte esterase. Leukocyte esterase is a white cell enzyme that looks for the presence of white cells in a urine. So these little screening tests could be done by the staff in the emergency room if they suspect UTI. And if those are positive, then at that point they may decide to collect the urine specimen and send it to the lab for a full urinalysis and a culture. So you want to make sure to mix the urine thoroughly before we streak the plates. And this is the only culture type that has a different streaking method than any of the other types of culture. So it's called a T-streak. And we do this, we would inoculate a urine specimen to a blood agar plate and then either the McConkey or the EMB, because remember they're interchangeable, use a calibrated loop. We're only going to work up cultures with 2 or fewer organisms present. So anything more than 2 organisms indicates that the collection was not clean. So it's an indication of contamination. A colony count of 10 or greater organisms, and we report that in CFU per milliliter, which stands for colony forming unit per milliliter. So colony count of 10 or more is considered significant unless it's a catheter specimen or some other type of sterile collection method. Then in those instances, any growth is considered significant. The colony count, and it's exactly what it sounds like. You're actually counting the colonies. And this is up to a certain point. Like if the plate is full from top to bottom, side to side, it's full everywhere with bacteria, I'm not going to sit and count every one of those colonies. You can kind of guesstimate it at that point. But that's essentially the idea behind the colony count. And the colony count is done only from the blood agar plate. Anything greater than 100 colonies is just reported as greater than 100,000. Plates are not held for longer than 24 hours, generally speaking, unless the doctor requests that for some reason. And then if we recover a pathogen from a urine culture, we're going to perform antimicrobial susceptibility testing. Rapid methods are most commonly used, and even the rapid methods sometimes can take like 12 to 16 hours. But antimicrobial susceptibility testing, we're just determining which antibiotics are effective at treating that infection. We report that to the doctor so they know how to begin treatment. So some common urinary pathogens that you could recover. So in the category of gram-negative rods, E. coli is probably the most recovered. It's antibiotic susceptible strains emanating from patients' fecal flora. Most common pathogen recovered in UTIs. Also some other organisms, Pseudomonas, Klebsiella, Acinetobacter, and Enterobacter. Those all are considered part of the group of organisms known as the Enterobacteriaceae, so we will talk about those more in this unit. Gram-positive cocci that are common causes of UTI. You've got Enterococcus, Staph saprophyticus, and if you remember from Unit 1, this is predominantly recovered in symptomatic, sexually active women younger than 40 years old. Strep A. galactiae. This one's, they say it's not as common, but I mean, I feel like I see it fairly regularly. But vaginal colonization can cause neonatal sepsis. You've got Staph aureus. According to the newer version of the textbook, a pure culture of Staph aureus should be considered significant regardless of the amount recovered. So, and pure culture, what that just means is that if Staph aureus is the only organism growing on the plate, then no matter how much, what the quantity is, we should consider it significant. And then Staph epidermidis is only seen in 20% of cases. And it's usually going to be in hospitalized patients over 50 years of age, usually who have had recent urinary tract surgery, have an indwelling catheter, or chronic urinary tract disease. Because again, Staph epidermidis is one of those organisms that we consider normal flora. So we might not normally think of it as a pathogen, but it could be in an immunocompromised patient. Some gram-positive rods that could be considered pathogens. And again, gram-positive rods, just kind of generally speaking, are one of those large categories of organisms that are usually considered contaminants. But there are a couple that could be pathogens. So Listeria monocytogenes. And this is just rare cases, and it's only considered significant after consultation with the physician, so maybe taking other information into account. And then Bacillus species. This is one of those that if we're isolating a Bacillus species, it can almost always be considered contamination. Some fungi, Candida albicans, which is rare to see in healthy adults, so it's more common in hospitalized patients. And early colonies, which means kind of like early on in the incubation process, can look similar to coag-negative staph, so their colony morphology can be similar. Cryptococcus neoformans, blastomyces, dermatitis, coccidioides immitis, and histoplasma capsulatum. Recovery of any of these organisms in urine culture is considered highly significant and indicates a disseminated infection. And then just some other kind of miscellaneous agents of UTI. Anaerobes would be significant if they are recovered from a suprapubic aspirate. Chlamydia trachomatis and Neisseria gonorrhea. These organisms can cause urethritis, cystitis, and prostatitis, so it can cause serious infection. However, these organisms would not necessarily routinely be recovered from a UTI. From a urine culture, and that's just because Chlamydia trachomatis, if you remember, is extremely hard to grow in the lab, and Neisseria gonorrhea only grows on modified Thayer Martin or chocolate or other specialty or enriched media. So we would either have to know, the doctor would have to request that we are looking for these organisms, or a better idea might be to do a urine specimen for molecular testing. Mycoplasma and ureaplasma. You'll see these in neonates from lower socioeconomic groups and potentially individuals with an HIV infection. And then Gardnerella vaginalis commonly reflects vaginal contamination, but it's also, in recent years, it's been emerging as a urinary tract pathogen. So if you are seeing repeated cultures from a patient where you've recovered this organism, then, and again, as the primary isolate, so the only organism growing, then that should be considered significant. For susceptibility testing, and again, that just means that we're testing the organism against certain antibiotics so that we can give a report to the doctor that shows them which drugs would be able to treat the infection. So Microscan and VITAC are the two most well-known rapid identification methods. This picture here is of a Microscan instrument setup. So you've got the instrument itself, the computer system and software that it comes with. This is a Microscan panel. So you can see each one of these little wells here. I know this picture is too small in this video, but each one of these wells is labeled. So these color reactions that you see up here are the different biochemical tests and like carbohydrate fermentation tests that the instrument is performing on this organism. So we inoculate all of these wells with like a mixture of the organism, and then the instrument reads these color reactions to get an organism ID. All of these down here are the different antibiotics, different dilutions of the antibiotics that the organism is being tested against, and that's how we determine the susceptibility. I'm going to let you read through these couple little slides here, just a few points to remember about urinary tract infections in general, and read through that case in point, just kind of a case study there, and just get you thinking about a couple different things. Okay, a couple review question here. Most common bacterial infection is a UTI. What's the only urine specimen type that would be appropriate for an anaerobic culture? And that's a suprapubic aspirate. And remember, that's the one where they stick the needle in through the belly, it goes to the bladder, and it pulls out the urine sample. If a urine culture grows 4 different organisms, and they're all greater than 100,000 CFUs per milliliter, what would be the appropriate course to take? So do we report the culture as probable contamination and request a new specimen? We're going to give identification and sensitivities on all of those organisms because they're all greater than 100,000. Report it out as normal flora. Give ID and sensitivities for only the gram-negative organisms. We're going to report that out as contamination and request a new specimen because remember, even though they've got a significant colony count, we've got more than 2 organisms. So when we've got a culture that has more than 2 organisms, that's considered it was not a clean catch, and it's contaminated. So that's the end of the first lecture for Unit 3.